Why this is important? For bacteria, universal B6 and B8 have conservative sequence. This is universal for all the bacteria. Therefore, you heard about the 16S-RNA biotechnology. 10 years ago, when I was students, this is the most fashion stuff in the world. Right now, everybody using these. That's where it comes from. Comes from 30th subunits. Okay? We'll mention this again when we talk about RNA translation. Nucleic acids, nucleus. This is simple. You have chromosome. Okay? Chromosome usually is like a closed circular double-stranded DNA. Supercoiled and the nuclear proteins aiding in the folding. And most of these proteins, most of these DNA is negative supercoiled because if positive supercoiled, the double layer, the double bonds is very easy to come back. Okay. But more importantly here, we want to talk about the plasmids. This is different from the chromosome. Plasmids, it's a very, we want to talk about this a little bit deeper. Let me just write up here. 3 key things for the plasmids. First of all, plasmids going to be independent or integrated into chromosome. But remember, when I say independent, which means their replication is independent. They do not rely on chromosome. But they can be integrated into the chromosome because they carry the genetic information, sometimes cause problems. Number 2, this is usually short 30 to 100 base pair sequence. Most of the time is linear. Okay? Number 3, plasmids is always cause problem, especially right now. Did you ever heard antibiotic resistance? When we talk about this, did you ever heard MRSA? You know that. If you're working in the WVU like a hospital. Methicillin-resistant Staphylococcus aureus. This is one of the higher risk stuff in the hospital caused by antibiotic resistance. One of the mechanisms behind is when we use antibiotics way too much, what will happen? Some of the plasmids will be released, and then they're going to carry on the signal or the information we call it all factor. This is related to antibiotic resistance. If they are coming to the environment, there is another bacteria, if they eat it, their chromosome could be integrated in there. Then the bacterial chromosome, if they have this off factor, this bacterial chromosome also become antibiotic resistant. Okay? So that's a problem. These plasmids is going through everywhere. And that could go through transformation, transduction, and conjugation to different types of the bacteria. And then they become universal antibiotic resistant or multiple drugs antibiotic resistant. It's all coming from these plasmids. Okay, so that is important. Just mentioned here, we could be mentioned detail later. External structure. Okay, we're going to talk about the villi, fimbrial, flagella. For villi and the fimbrial, we'll just talk briefly. Okay, these are very short, thin, I will say hair-like appendages. They are for attachments, motility, DNA uptake, especially for the sex villi. and we say it's required for conjugation. I will say for phyli and fimbrial, the more important is for transfer genetic information. Motility and attachment a little bit, not really as important as capsule and flagella. Okay, so very brief, we can read through the slides. But for flagella is more important. We will do in the lab for the flagella test. Okay, so this is we want to talk a little bit more. First of all, flagella composed by flagellin, we mentioned, a protein, very soft, so we have a flagella skin, a very special skin. Second, if bacteria have flagella, that's what we call real movement. Nozzles tumbling. Okay? How come you have a real movement? Because there are different categories. Some of the bacteria only have one tail at the ends. Let's say Pseudomonas. We will talk about this in the lab all the time. Pseudomonas aeruginosa. This is what we call monotrichos. Monotrichos. Second category, you have a bacteria on the two ends called Anthitrachus. For example, Staphylococcus aureus. Then you have some of the bacteria will have a flagella stacked on one end. Liphotrichos or Lophotrichos. I don't know the good example, maybe vibro, maybe. The last one. This is everybody knows, we will mention in the lab again and again. Proteus vulgaris. The flagella covers the whole surface and everywhere. And I mentioned maybe there's no colony forming. It's called a swamp. This is called a petritrichus. Okay, that's on the four different types of the category for flagella. If the bacteria has petritrichus, which means they have a very, very strong movement motility. Look at this C here, Proteus vulgaris. It's a petritrichus flagellum. It's everywhere, covering the surface. We don't mention archaea too much, but we are going to mention spirochetes. Is that right? Spirochetes are very special bacteria, and very soft. They have a very special cell structure. And we mentioned one particular pathogen called Treponema pallidum, which is called Syphilis. What it looks like, they don't have a flagella. Their flagella a little bit different, which is called corkscrew-like movement because they have endoflagella. So their flagella a little bit different. We will mention this again. Now, why we say flagella have a real movement? When you look at the slides, this is for bacteria is like taking a taxi. It's round trip taxi. Okay, we use a lot before pandemic. We don't use that anymore, I believe so. Chemotaxis, what that means. If you have a signal somewhere for bacteria, it's either attracted or repelled, the bacteria will go there and then they will be flee away. It's a round trip. Now why they can do a round trip? Because your cell membrane system have a sensor, can sensoring these signals. And then why they can do a movement? Because they have a flagella. How fast they can do, how frequency they are going to be reacted to the signal depends on the structure and how many flagella they have. Okay, so this is called chemotaxis. Very simple, we mentioned this. Okay, last one. We want to mention endospore. We want to talk about this before we end up today. This we have some things we could talk. Endospore. Okay, first of all, you already know they're the endospore stem. We will practice on Thursday. And this endospore stem, you need to use heat steeping. Okay, then the question is why we need to do that. So, second question, what other bacteria has endospore? Basically, Bacillus. Bacillus, we'll talk about Bacillus subtilis and Bacillus cereus and Clostridium. You heard about the Clostridium all the time. Clostridium perfringens, Clostridium difficile, Clostridium tetanus. These two major genera names has endospore. Usually, in a normal growth situation, they will just grow. Okay? How about harsh environments? Or we say stress environments. They will generate endospore. We call it sporation. And when the condition changes, when they come from a harsh environment to a friendly environment, they will come back and become vegetative cells. Then we call it germination. Now, this sporation could be very long, weeks to years, or maybe 100 years. Okay? This is the first thing. In a very harsh environment. Now, second thing, we want to give you an example. What kind of situation will have windows? This is a real story. Back like 20 years ago, somebody shared with me. I'm not that old, so I don't experience that. I used to work in Western Kentucky University back in near the border of Kentucky and Tennessee. One of our faculty members, there is a friend who's working University of Louisville. One day, his wife is cooking something. They do low temperature and a long time cooking. Okay, a soup. What happened for this soup? The soup, they put some carrots there. They put some beef stew there. Or probably put some tomato there. Okay, there's the problem. The beef stew, when you put in the soup, it becomes a beef broth. It's a bacterial media. When you look at the label of the bacterial media, it's a beef broth or beef extract. Carrots or tomato, where they come from? Soil, is that right? Where going to be the endospore originally come from? Come from soil. So once they do low temperature, long time cooking, might be like a 50 degrees Celsius, you can say that, maybe like 8 hours. Then what happens? All the normal bacteria will be killed. Then the endospore bacteria could be Clostridium, could be Bacillus or something else, will be grow dramatically and dominate the soup. So one day their family go to hiking for a whole day. You know, in the Kentucky area, there are lots of the mountains. You can go hiking the whole day. When they come back, then the husband said, okay, let's everybody have a soup. Then the wife has one, the son has one, the dog has one. Just like 4 hours later, everybody has vomiting and diarrhea, and they go to university or Louisville Medical Center. Some of them go to medical school there. Why? Because all these generate endospore have intoxication infection that could happen within 4 to 6 hours. Okay? So that's a story tells you the low temperature, which is actually a very harsh environment, stress environment for bacteria to survive, then for the endospore to generate, and then the normal bacteria will be killed. Okay, so this is what happened for the endospore. Now the last question. Why the endospore will be protected in these harsh environments? If you learn the endospore structure, we're going to talk about these. Cortex, okay, coat, center. There is a very special called calcium, big, pearl, formic acid, and small soluble acid DNA. Once those two combined, it's become a very special chemical structure to let the bacteria can protect the harsh environments. Okay, now we know we can do endospore stem. Why we do it? Because we know the position. Could be terminal, subterminal, and in the center. Last thing, endospore is a protection structure. There is no reproductive function, which means a mother cell has an endospore. Does not mean the daughter cell will automatically have. The reason is, if the daughter cell is in a friendly environment, it will not form endospore. If the daughter cell also in a harsh environment, it may not have a sporation at all. So it's a protection structure. It's not a reproductive structure. Okay? So that's all I have for this section. And we'll see you Thursday.